I am Leema Rosemary, Assistant Professor, St.Joseph College of Arts and Science, Cuddalore. Now I am going to talk about structure of muscles and muscle contraction. Muscles are bundles of elastic fiber. They do all kind of work by contraction and relaxation. Muscles are known as effectors. It made up of myocytes or muscle fibers. Human body has more than 600 muscles. Functions of muscles. The muscle cell functions like biological machines that converts chemical energy into mechanical work. The mechanical work involves the various movements of contraction of heart and blood vessel. Muscle is an excitable tissue responsible for most of our functions like locomotion, posture, speech, respiration, pumping of blood, movement of food, expulsions of waste, and storage of energy. Types of muscles. The muscles of the vertebrates are classified into different groups depending upon presence or absence of cross striations. They are striated muscles and non-striated muscles. Depending upon the control, the muscle can be classified into two groups: voluntary muscles and involuntary muscles. Depending upon the situation, the muscles can be classified into three types: skeletal muscles, cardiac muscles, and smooth muscles. Striated or stripped muscles. It is the muscles which has a large number of cross striations. Skeletal muscles and cardiac muscles comes under this category. It's our voluntary muscles. They are obeying the will of the individual, since the stripped muscles are found in association with skeletal system. So they are called skeletal muscles. Example: triceps, biceps, etc. Then non-striated or unstripped muscles. Muscles which does not have any cross striations. It also known as plain muscles or smooth muscles, and functionally it's an involuntary muscles. It is found in wall of the visceral organ. They have the connection with the internal parts of the body. These are generally forming the walls of the stomach, intestine, blood vessels, ureter, etc. Then cardiac muscles is specific for heart. It is generally intermediate in structure between the striated and non-striated muscles. Function as a involuntary muscle. This is the structure of muscles. This is composition of skeletal muscles. Muscles contains water, 75% water and 25% solids. The solid contents are organic substances, inorganic substances. Organic substances include proteins, myosin, actin, tropomyosin, troponin, actin, desmin, nuclein, myoglobin. Then other organic substances are carbohydrate, lipids, nitrogenous substances like urea, uric acid, xanthine, hypoxanthine, creatinine, phosphocreatinine, etc. Then inorganic substances: potassium, sodium, calcium, magnesium, chloride, phosphate, sulfate. Potassium is the principal mineral in the muscles. This is the structure of skeletal muscles. They comprise the main bulk of the body mass of the vertebrates. Each muscles bear at the end of the fibrous cord, that is tendon. The tendon used to attach the muscle with bone. Each muscle is enveloped by a connective tissue layer called epimysium. Each muscle is made up of large number of muscle cells. These cells are called muscle fiber. Each muscle fiber is covered by a thin connective tissues called endomysium. The group of muscle fibers form bundles called fasciculi. This is a fasciculi. It's covered by connective tissue called perimysium. This is perimysium. Then several fasciculi unite to form a large muscles such as triceps or biceps. The plasma membrane of muscle fiber is called sarcolemma, and the cytoplasm is sarcoplasm. So, sarcoplasm contains numerous longitudinal protein filaments called myofibril. Each myofibril is composed of a number of alternating light band and dark band. A sarcomere is the basic unit of myofibril. It lies between two Z line. It is made up of a central dark band and two half light band. A sarcomere consists of two kinds of longitudinal filaments, namely thick filaments and thin filaments. This is the structure of muscles. This is the myofibril. This is dark band. This is the light band. The structural unit of myofibril, that is sarcomere. This is the myofibril, is a dark band. This is light band. The middle line is Z line. So, two Z interspace between the two Z line, that is sarcomere. So, Z line, this is Z line, center dark band, two half of light band. This is a sarcomere. The thin filaments are attached to the Z line at one end. Their other end extend towards the middle of the sarcomere and are free. The thick filaments lie midway between Z line and are unattached to them. The region of thick filaments represent the dark bands. In the middle of the dark band, the thin filaments are absent. The region of the dark band where the thin filaments are absent represents the H zone. The region of the thin filaments constitute the light bands. The next, contractile elements of muscles, that is protein present involved in the muscle contraction. So, muscle is composed of two filaments, namely actin filament and myosin filament. Myosin filaments are formed by myosin molecule. Actin filaments are formed by three types of protein: actin, tropomyosin and troponin. These four proteins together constitute the muscle proteins or contractile elements of the muscle. This is the myosin filament or thick filament. A thick filament is composed of a bundle of protein molecules called myosin. Myosin molecule is rod-like. It consists of a head, a neck and a tail. The head is also called cross bridge as it attached with actin during muscle contraction. Each myosin filament consists of about 200 myosin molecule. Then each myosin molecule made up of 6 polypeptide chain, of which 2 are heavy chains and 2 are light chains. The total molecular weight of each myosin molecule is 4,80,000. This is the structure of myosin. It looks like a gold stick. This is the head portion. This is the tail region. This is the myosin filament. That is 200 myosin molecules present in this thick filament. This is the structure of myosin. It's 2 heavy chains, 4 light chains. The heavy chain twists around each other to form a double helix. At the other end, both the chains turn away in opposite direction and form the globular head portion. To each part of this head attached to 2 light chain. This side 2 light chain, this side 2 light chain. So, head portion to form 4 light chains. Each myosin head has 2 attachment sites. One side is for actin filament and the other side is for an ATP molecule. The next, thin filaments. So, thin filaments made up of actin, tropomyosin and troponin. Actin molecules are globular and arranged in the form of a double helix. These are actin molecule. Then tropomyosin. These are filamentous structure. It lies in the grooves of twisted actin strand. Then next, troponin molecule. It is nearly oval in shape. It is associated with the end of each tropomyosin molecule that lies site that lies towards the center of the sarcomere. So actin filament. So actin molecules are the major constants of the thin actin filaments. So each actin molecule is called F-actin, and it is the polymer of a small protein known as G-actin. This is a F-actin. It formed from the G-actin. There are 300 to 400 actin molecules to form the actin filament. In each F-actin molecule has an active site to which the myosin head is attached. That is, actin filament contains only one attachment site. This is the G-actin, this is the F-actin. That is, the globular actin made to form double helix, that is F-actin. This is the structure of tropomyosin, filamentous structures. This is oval shaped troponin. So these three form the actin filament, that is thin filament. Then tropomyosin. Tropomyosin, there are about 40 to 60. The tropomyosin molecule situated along the double helix strand of actin filament. Each tropomyosin molecule has the molecular weight of 70,000. In relaxed condition of the muscle, the tropomyosin molecules cover all the active sites of the F-actin molecule. Then troponin. So troponin is one of the actin filament. This is formed by three subunits, troponin I, T, C. So actually I attached to F-actin, T attached to tropomyosin, C attached to calcium ions. So totally troponin contains three subunits, that is T, C and I. Then muscle contraction. So muscle contractions means shortening of muscles. That is the process by which muscles are shortening. Muscle contract in response to various stimuli such as mechanical, electrical and thermal. There are two types of muscle contraction, that is isometric contraction and isotonic contractions. So isometric contractions means, iso means same, metric means length. Here the contracting muscle retain its original length. This type of contraction is called isometric contractions. Here the maintaining the same length. Tension developed but not able to move a load. For example, when we have a hand against the wall, muscle tonus is increased, that is tension is developed but no mechanical work done. This is the isometric contraction, same length. Then isotonic contractions, here change in length and also tension developed, that is work done. The muscle is able to move a load and it shortening during contraction. This is called isotonic contraction. The best example, walking, but in running is the example for both isotonic and isometric contraction. Then theories of muscular contraction. Electrokinematic theory. This theory is not accepted. This theory explains that the myosin filaments carry series of negative charge, actin filament carries series of neutral charge. In the resting condition, there is no electrostatic attraction between these two types of filament. But activation, calcium ions are released into the cytoplasm from the sarcoplasmic reticulum. So the released calcium ions make the actin filaments to positive. So the positive charged and negatively charged myosin. So the positively charged actin and negatively charged myosin filament are attract one another. Hence the actin filament slide to over the myosin filament. This theory is accepted. this theory is not accepted. Then another theory, sliding filament theory, that is sliding mechanism and formation of actomyosin complex, otherwise sliding theory or ratchet theory or walk along theory. So many names. First, the theory was proposed by Huxley and Hansen. It is based on the structural changes occurring in the muscle fiber during contraction. Sliding theory explains how the actin filaments slide over myosin filaments and form the actomyosin complex during muscle contraction. Then each cross bridge from the myosin filaments after binding with active site of F-actin, the myosin head is tilted towards the arm so that the actin filament is tagged along with it. This tilting of head is called power stroke. After tilting, the head immediately break away from the active site and return to original position. Now it combines with a new active site on the actin molecules and tilting movement occurs again. Thus, the head of cross bridge bent back and center of sarcomere. In this way, all the actin filaments of both the ends of sarcomere are pulled. So, the actin filaments of opposite sides overlap and form actomyosin complex. The formation of actomyosin complex results in contraction of the muscles. The events of muscle contraction. Actually, this is the resting state. This is the activation state. Here, myosin molecule attached with ATP. This is the actin filament, no attachment. But here, the ATP hydrolyzed into ADP and inorganic phosphate. So, myosin head ready to bind with actin. Here the myosin bind with actin, that is actomyosin complex. Then produced power stroke. So, the myosin head towards the actin filament. Here the ADP and the inorganic phosphate removed as a ATP. Here the ATP again it bind with myosin head. So, the attached actomyosin complex detached after binding the ATP. Thank you.